Okay, so that's the cytoplasm. We talked about the plasma membrane, and then the cytoplasm is everything that's inside the plasma membrane that's not the nucleus. The last thing we have to talk about is the nucleus. We're not going to go into a huge amount of detail. What we are mostly concerned with is identifying the nucleus in a cell so that we can help figure out where one cell stops and another cell starts when we look under the microscope at tissues in lab. So the nucleus is the control center of the cell. It's where the DNA is housed and stored and protected. It's the reason we have a nucleus. It's basically a space in the cell that's separated by phospholipid bilayers from the rest of the cytoplasm. It's a calm, stable environment for the DNA. The DNA is the blueprint for all of our proteins. The DNA is used to make RNA. The RNA is read by ribosomes to make proteins, but the DNA is the master copy of the instructions for how to build each protein in your cells. That needs to be kept separate and safe and in a nice, stable environment. And that's what the nucleus is. It's a nice, stable environment to keep the DNA in, and also to regulate which genes, which sequences of DNA are used to make proteins in which cells. Different cells use some genes, and then other cells use other genes. You have about 20,000 genes in each of your cells, but each of your cells don't use all 20,000 of those genes. And so some types of, like epithelial cells, might use one set of proteins. Muscle cells might use a different set of proteins. Neurons might use another set, and so on. So the nucleus is where we figure out which proteins a cell is going to use, and therefore what type of cell it is. And usually it's about 5 micrometers in diameter, so smaller than a typical size of a cell is maybe anywhere from, say, 10 to 50 micrometers, millionths of a meter in diameter. And so the nucleus is smaller than that. It's roughly 5 micrometers in diameter. And so the boundary of the nucleus is called the nuclear envelope. And it's actually 2 phospholipid bilayers with a space in between the 2 phospholipid bilayers. And that space is continuous with the lumen of the rough endoplasmic reticulum. And sometimes you actually see ribosomes on the outer phospholipid bilayer of the nuclear envelope. And sometimes proteins are secreted in that space between the 2 phospholipid bilayers of the nuclear envelope. Proteins that are going to be secreted are made in that space. The lumen in between the 2 phospholipid bilayers of the nuclear envelope is kind of the same and has the same functions as the lumen of the rough endoplasmic reticulum. To get from the nucleus to the cytoplasm, you need a pore or a tunnel to go through the nuclear envelope. And so the nuclear pores are those tunnels, and they're made of big proteins, multiple proteins that cluster together and form a tube that go through both phospholipid bilayers of the nuclear envelope. And the tube is big enough so that big molecules can get in and out. Proteins that regulate DNA can go from the cytoplasm into the nucleus. And then RNA that's made using the DNA in the nucleus as a template can leave the nucleus and go out into the cytoplasm and be read by ribosomes to make proteins. So these nuclear pores are big. They let big molecules go in and out of the nucleus. And then inside the nucleus, there's usually a structure called the nucleolus, which stains on using most stains, it stains a little bit more darkly, or it looks more dark than the rest of the nucleus. And that's the part of the nucleus, that's where the DNA that's used to make more ribosomes is found. And it just tends to look a little bit more dark. And sometimes in some cells, the nucleolus can be a hint that you're looking at a particular type of cell. So the nucleolus is really visible in multipolar neurons in the spinal cord, for example. So when we look at those cells and we see that little dark spot in the nucleus, that's a hint to us that we're looking at a motor neuron, a multipolar motor neuron in the spinal cord. And don't worry if that doesn't make sense to you yet, but the idea is by recognizing the nucleolus, we can get an idea of what specific cell we're looking at in some cases. So the nucleolus is where what is made. what's made by the nucleolus. What's the significance of the nucleolus in a cell? It's where ribosomes are made. Yeah, exactly. Okay. And here's our picture of the nucleus. We can see there's a phospholipid bilayer here and here, and then there's a space in between. And you can see that space is continuous with the lumen of the rough endoplasmic reticulum right here. And then you can also see there are these pores, these orange pores that go through both phospholipid bilayers and let big molecules go out or in. RNA molecules go out so they can be read by ribosomes in the cytoplasm. Proteins go in to regulate the DNA and determine which genes are used in a particular cell. And here we can see the nuclear pores from the surface. And again, just to be specific, the name of the membrane, of the structure that surrounds the nucleus, is called the nuclear envelope. And we don't really say nuclear membrane because it's actually two membranes. So nuclear envelope is kind of a special term because it's two phospholipid bilayers, not just one, like the plasma membrane. Okay, and then that's that. So I know it seems like it's just a deluge of information, especially if you haven't had a ton of cell biology before. So if you're feeling panicked, one thing I'll say is mostly for us in anatomy, what you need to remember from this lecture can be summarized by the big table in chapter 2. And in the 8th edition, I'm going to just open my book and find that table for you. So if you're feeling panicked by all this information, if you've never had a biology course that's dealt with this stuff, in the 8th edition, table 2.1 basically just summarizes most of what I've said, and don't panic, learn what's in that table, and that's going to be like 80% of anything I'll ever ask you about this chapter on the test. And so then you might be saying, well, why did you go into detail on the other stuff? The reason is because you're going to need to know all this and more in physiology, and it's going to come at you twice as fast. And if you've heard it once before, it's going to help you out. So learn as much as you can of what I've just said. And if you're panicking, focus on the material in table 2.1. And that's the core, the essence of what you need to know for the test for this class.